Thank you for PMWC and FDNA for inviting me here. In terms of disclosures, I have no financial disclosures, but I am on the scientific board of the Face2Gene software development program, which I'm going to be discussing today. So let me just find the clicker. What I'm going to be speaking to you today about is next generation phenotyping. And hopefully by the end of this talk, you'll share some of the enthusiasm and passion that I have for the evolution of phenotyping that is evolving around us. The first question we should ask ourselves is, what is next generation phenotyping? Deep phenotyping has been around at least since the 1950s, when a group of clinicians who practiced in rare and undiagnosed diseases, specifically structural malformations, developed a discipline called dysmorphology. The use of specifically annotating the minutiae of a phenotype really is something that's been done for a long time. So how do we differentiate that from what we think of as next generation phenotyping? And really what it comes down to is, where do we limit our thoughts as to what a phenotype represents? Is it limited to structural morphology, or do we consider all aspects of a phenotype in the context of what's going on? Just thinking about speech analysis, gait analysis, metabolome, microbiome, any of the omics. Yesterday I heard Dr. Olivia Walsh talk about her work in circadian rhythms, and it taught me about a new omics, chronomics. So as we develop all of this big data, one of the big things that we're learning with machine learning and deep learning is, where do we limit ourselves? And I think that's what we have to look at it as the next iteration. So I would argue that if we think about a phenotype, it's anything that is not related to a genotype. Part of the problem is that we generally don't speak a common language. So one of the things that the dysmorphology group did, in around 2009, the NHGRI put together a website and a portal that allows people to speak a common language. So the development of the human phenotype ontology, which really develops a hierarchical ontology that allows people to very specifically define structural phenotypic abnormalities. Where does this lead us to? In terms of human development, it gives us a basis to describe specific developmental abnormalities. One of the really phenomenal initiatives that has evolved out of all of this big data is something called the Monarch Initiative. And the Monarch Initiative is really an open source collaboration between a number of different organizations that aim to link genotype to phenotype in an effort to advance precision medicine. Helps associate developmental abnormalities across species so that genetic pathways can be better identified, that disease mechanisms can be better defined. One of the reasons that I am particularly excited about working with Face2Gene and actually utilizing Face2Gene is that it's a methodology that bypasses some of the biases that we intrinsically have when we walk into clinic. I don't know how many clinical geneticists there are in the room, or dysmorphologists, but we tend to have a little bit of pride and honor about our abilities in terms of diagnosing dysmorphic disorders. What Face2Gene allows you to do is support that decision-making process. And the beauty is that it's in a portable handheld device that you can take anywhere with you. And the full functionality you can then utilize once you're back at your desktop. The essence is that it's not looking at dysmorphology. It's looking at data points and really taking a number of data points across the facial characteristics and creating a digitized fingerprint or footprint of those anthropometric measurements. One of the biggest challenges we had when we thought about integrating this into our clinical workflow was privacy. We all know about HIPAA, about the strong concerns about sharing HPI. And one of the things that we had to assure ourselves was, how can we justify taking a clinical image of a patient, sharing that with a vendor, and yet being able to utilize the freely accessible clinical utility that it offers. And fortunately, Face2Gene thought about that. I'm going to run you through the process fairly quickly in terms of what happens when you utilize the software. Obviously, the first step is to take a picture of the patient, put that into a secured portal that only the user has access to. The vendor doesn't have access to this portion of the portal. What then happens is that there's a digital signature that's extracted from that image based on those anthropometric measurements. That then gets fed down into essentially a machine learning tool, and I'm not going to try and speak about the math because Jenna would put me to shame here. And then using that deep learning and machine learning capability of the tool, present the end user with a differential diagnosis which can then be used to support the clinical expertise of the person in clinic. Because this is really a crowdsourced deep learning, machine learning tool, it relies on people feeding the information back into the system. And as such, I encourage all of the people that I work with to both utilize the tool, but once we have confirmed a molecular diagnosis, to go back in and tell the machine that it was correct or what the accurate diagnosis was, because that'll just help the next person who's using the tool. So I'm talking about accurate phenotyping. How important is accurate phenotyping, and why am I so excited about having a tool that doesn't rely on me describing characteristics? I thought I'd run you through a quick example of a well-recognized phenotype. So what we have is somebody who has microcephaly, simple cupped ears, hypotelorism, in other words, closely spaced eyes, a broad-based nose with a prominent tip, and a relatively wide mouth. So just out of curiosity, how many dysmorphologists are in the room? None. So I'm sure everybody had this in their mind, Mr. Potato Head. All right. So we have all of these great tools available to us. I don't know how many of these you're familiar with, but in the context of rare and undiagnosed diseases, we try and utilize all available tools that we can use to improve our diagnostic acumen. All of these rely on us defining HPO terms and putting them into a system, and then it feeds information back to us. And as you can see, we can get a variable feedback based on how far down that hierarchy on the ontological path we go. The more specific features we have, the more targeted we could be. One of the really fantastic things about using a tool like Face2Gene is just how reliable it is with some of the more recognizable disorders. This was a study that was performed by a group who took a very specific, well-recognized phenotype. So this individual has a disorder called Cornelia de Lange syndrome. You can essentially walk into a baby's room and make this diagnosis with not too much difficulty. What they then did was they set up a collage of patients with known diagnoses, so known patients with molecularly confirmed Cornelia de Lange syndrome, and some patients who had phenotypically similar disorders, but not that diagnosis. This was then presented to a panel of experts, and they were asked to correctly identify those patients that had Cornelia de Lange syndrome and those patients that did not have Cornelia de Lange syndrome. And the experts were able to positively identify 77% of the time, and they were able to recognize the phenocopies 87% of the time. That gives us a clinical sensitivity of around 82% and a specificity of around 89%. When the software was challenged with the same question, a positive diagnosis was recognized 94% of the time, and 100% of the phenocopies were identified. That gives us a clinical sensitivity of 86% and a specificity of 100%. Now, this is a clear, well-recognized, obvious syndrome. So there was a publication recently, which I thought was quite daring of them. In March 2018, this group published this disorder, where they took a really complex group of disorders, essentially children with inborn errors of metabolism who have mild coarsening of their facial features. They added in the NCBRS, which is the Nicolaides-Baraitser syndrome, because the phenotype is similar to patients who have storage disorders. So we have Smith-Lemli-Opitz syndrome, mucolipidosis, mucopolysaccharidosis type 1, and mucopolysaccharidosis type 2. And when just looking at pure facial imaging, the software was able to accurately predict the specific disorder in 64% of the cases. If you added a single feature into that, a feature set, the diagnostic accuracy increased to 87%. So in complex disorders, it's still able to bring that diagnosis to the fore and help you think about a clinical diagnostic testing to proceed with. If we think about overall performance analysis, this tool has now been available for almost 6 years, and the data that's been collected over time has now reached a point where we can actually look at it and say, well, how well does it perform as an average? And if we take the top 250 syndromes that occur most commonly, the area under the curve averages around 98%. And what does that mean? An area under the curve of 50% means that it's a random chance association. So we can see that this has a really high average performance, actually across hundreds of syndromes, which then enhances the clinical utility of the tool in the clinic. One of the things that struck me yesterday when Dr. Collins was talking about data in his keynote speech yesterday was that he mentioned that as data accumulates, we become better and better. And really, that speaks to this tool too. The more we use it, the better it gets. I'm really comfortable looking at the urea cycle and thinking about all the disorders that are associated with that. And as I said earlier, I'll leave this to Jenna, because convoluted neural networks is definitely not my forte. But I think it's a really important thing to think about, and especially when it comes to thinking about deep learning. And really, it's the function of sharing, utilizing a tool, and looking at this next generation phenotyping and asking ourselves the question, where are the limits? Does it have to be limited to what we know now? Could we use any data that is not predominantly genomic and feed it into these essentially AI tools to help us in what we're trying to do? All right. Thank you.